Hello guys, it's Coach Rich. We're going to go over these notes a little bit different today. I'm going to use software called Screencast-O-Matic. And in doing so, I'm going to walk you through your notes, talk to you a little bit about genetics, and see what we can do here. Okay, so let me pull this up real quick. If you have any questions along the way, just let me know. So we're going to start here. You should have your notes out. We're going to start at the top with number 1, genes and chromosomes. But first, let's talk about genetics. So, genetics, as we all know, is the science that studies how genes are transmitted from one generation to the next. So, your parents pass their genes on to you, and you in turn will pass your genes on to your offspring, and so forth and so on. So go ahead and write this definition down for genetics, as I will do the same. If at any time you need to pause this video to finish your writing, that is perfectly fine. Please do not skip ahead because you're going to want to hear what I have to say at some point. And if not, don't skip ahead just simply because I ask you not to. All right, so I'll move on as soon as I get things written down. I'm taking these notes on my right side of my computer, just like you are. Okay, remember we did this. This is called Hitchhiker's Thumb. This is a giant sequoia tree in California, and these are different types of corn here. Obviously, you can see the genes in this corn is very different from one another. Okay, starting with genes and chromosomes now. The chromosomes. We all know what chromosomes are. We have 23 pairs of chromosomes. 46 total, 23 from mom, 23 from dad. 22 pairs of those are called autosomes, and 1 pair of sex cells. So our chromosomes are contained or found in the nucleus of our cells. They are made up of 2 things. I'm sorry. I got ahead of myself. I was thinking about nucleic acids there. Chromosomes are made up of DNA. It's a condensed version of DNA. And on that DNA, we find genes. We've already talked about a gene, but it is a short segment of DNA that controls a trait, and specifically a hereditary trait, something that you get from your parents. So go ahead and write that definition of gene there for me. So we have chromosomes, and in those chromosomes we have what is DNA, and then on the DNA, as you can see here, we have a gene. And at some points, I will go ahead and write on this slide for you if I have anything I want to tell you. I can take notes here as well, but we're not going to do that just yet, okay? All right, moving on. We know that chromosomes, we've talked about these, are a long chain of genes. Again, a chromosome is a large body of hereditary material. In that chromosome is a lot of condensed DNA. We talked about that being wrapped around histones, forming a nucleosome, and those nucleosomes forming coils and supercoils to tightly pack inside of there, okay? And then on that DNA, or on that DNA, we have short segments of genes, just like this, that allow for our proteins to be made, and specific proteins give us our traits, which takes us to trait. Let's go ahead and write this down. It is the characteristics that an organism has, such as hair color, eye color, tall or short, skin color, etc. So traits are obviously what we look like, how we act, and a trait is what we call a phenotype. It's our appearance, our how we act. It's our trait. Okay, and then those are coded for by our genes, specifically our DNA, and even larger than that, our chromosomes. Okay. So we haven't heard this word before, but it's one of your definitions. It's two alleles, so we're talking about alleles now. That is a new word for you. Must be present in order for a trait to show up in the offspring. And this is kind of like chromosomes here. One allele comes from mom and one from dad. And this is what we use to make Punnett squares here. So when fertilization occurs, an egg forms or fuses with the sperm cell and forms a zygote, the new offspring then has two alleles for that trait. So for instance, your eye color, you're going to have two alleles for that. Okay, and depending on what those two alleles are, the two you get from mom, the two you get, depends on what the offspring is. Okay, so you'll have two alleles, and then obviously what happens if you have children, you'll have two alleles. Your wife or husband will have two alleles, and then when egg and sperm fuse, those two alleles will mix, and depending on how those two alleles combine will depend on the trait that your offspring has. Usually it's some form of what the mother or father has. Sometimes it can be an in-between or a mix if it's codominant. All right, so this guy is known as the father of genetics. His name is Gregor Mendel. He was an Austrian monk. Austrian, not Australia. Born in 1882. So go ahead and write this down for me for number 1. 1822. Guys, that's not too long ago. It's less than 200 years ago that we started learning about all this stuff, and now we can do so many things. He's known as the father of genetics. He coined the term genetics. And he discovered the 3 laws, which we're going to talk about. The 3 laws of genetics that forever changed biology. We still use these laws today. They are laws. They are certain, unlike a theory, which can't be proved. Laws are thought to be universal. He conducted series of experiments in a quiet monastery garden. He did so using pea plants. Spent about 14 years growing these pea plants and experimenting with them in this garden. It's another picture of Gregor Mendel. He gave us 3 basic laws, and we need to write these down. Remember, a law is something that is universally thought to be true. It's unlike a theory. Theories cannot be proved. Laws are. So let's write these down. Number 1, the law of dominance and recessiveness. We'll talk about that in a little while. Number 2, principle of segregation. These are some of your vocab words as well. And then independent assortments. And Mendel's greatest contribution to the world of genetics was that he proved, remember we said genes in our test had to be able to carry information and transfer that information from mother or father to offspring. Well, he proved that inherited characteristics are carried by genes. So go ahead and write that down. Inherited characteristics are carried by genes. Let's underline the word genes. And inherited characteristics. So our inherited characteristics are carried by our genes, excuse me. That is our genotype, and those are demonstrated as traits in what is known as our phenotype. Okay, so why did Mendel choose to do these experiments with pea plants? He did so for a couple reasons. Number one, let's go ahead and write this down for B. They were readily available at that time. Peas were a crop that was easily obtained. They grew very easily. Remember, these things would have taken years and years to figure out if he was working with humans. But instead, peas grow very, very rapidly. So number 3, they grew rapidly. So he chose pea plants because he could get his hands on them. They grew easily, didn't need a whole lot of special, a real large specified amount of anything, and then they grew really fast. And he also chose them because you could see things on them very easily. So the sexual structures of the flower, so pea plants do have a flower. If you look down here, this pink flower or bloom, are completely enclosed within the petals so that there would be no, and we'll talk about this in a little while, no accidental cross-pollination. So he wanted to make sure when plants reproduce, generally they're blown, the seeds and things are blown through the wind. Pollen, the male gamete of plants is usually blown through the wind, lands on the female part of the plant, fertilizes it, and that's how we get new plants or seeds. In turn, he wanted to make sure that he could know for certain which plants were breeding with which plants. So he could control the breeding patterns of these pea plants, which was very important. So before we learn about Mendel's experiments, we do need to review the basic parts of a flowering plant. They contain both male and female reproductive structures. We call the female part of the flower the pistil, and it produces egg cells. The male part of the flower is called the stamen, and like I said earlier, it produces pollen, which contains sperm cells. We all know what pollen is. If you have any allergies at all, you're probably a little bit allergic to pollen. That's what's so high in the spring and what causes everybody's allergies to get really messed up. So when the pollen's delivered to the pistil, as you can see here, here's the pollen from another plant delivered to the pistil. Sperm travels to the egg cell, and the result is a zygote or fertilization. And then fertilization, again, like I just said, produces a zygote or a tiny embryo. Which is inside the seed. All right. So, how did he use these pea plants? So, anytime you talk about genetics, of course, you're going to have to talk about Mendel. Okay, so this is the most boring part of genetics here, but we do need to learn a little bit about history as we do these things. So pea flowers are normally self-pollinating. Since male and female reproductive structures are really close, generally the sperm from one flower fertilizes the egg of the same flower, so that's what we call self-pollinating. The resulting embryos have the same characteristics of the parent, which makes sense because the parent is the child, so to speak. The parent has the kid. So it's not asexual reproduction, but it resembles asexual reproduction in the sense that it's genetically very, very, very similar with respect to DNA, unless there's some mutations involved. So even though there's sexual reproduction has occurred, there's just one parent. It's what we call self-pollinating. Well, this guy Mendel, who was wise above beyond his years, knew that these pea plants were true breeding. And what does true breeding mean? That means that if he allowed them to self-pollinate, they would produce offspring identical to themselves. Again, true breeding. You may see that word again. For example, if we allowed to self-pollinate, tall plants would always produce tall plants. With yellow seeds, or plants with yellow seeds would always produce other plants with yellow seeds. True breeding plants were the cornerstone of Mendel's experiments. So Mendel wanted to produce seeds by joining the egg and the sperm from two different plants. So he wanted to control this breeding. So to do this, he again, guys, he was wise beyond his years. He had to first prevent the possibility of self-pollination. So he had to prevent the possibility of the sperm from one flower getting to the egg or the pistil of the same flower. So as we look here, we have the male parts, we have the female parts, so we have the stamen, the pistil on the stamen is pollen. So we didn't want that to happen, okay? So what he did is he manually cross-pollinated these plants. So he cut away the stamens. He took the stamens off the plant so that there was no male reproductive parts on the flower. Then he dusted the remaining female structure with pollen from a different plant. You can kind of see that here. He took the pollen from one plant and inserted it into the stamen of another plant. And this technique was known as cross-pollination, which produced, obviously, two different parents and offsprings. Well, it was produced, the offspring was produced from two different parents. So this is kind of what we think of as sexual reproduction, although technically self-pollination is sexual reproduction, but it's same parent producing offspring. So now Mendel was able to crossbreed these plants, so he could study characteristics. If he crossbred a red seed plant with a yellow seed plant, then he could kind of see the difference, the offspring or what the offspring produced, and he can make judgments based on that, okay? So we've already done this. Which thumb do you put on top? So go ahead and do that one time. I put my right thumb on top. Person in the picture obviously puts their left thumb on top. Before we proceed, I do want you to become familiar with a couple terms, and that is P generation, and that is what we call the parental generation. So that is the alleles or the genotype or genes from the parents. F1 offspring is the first generation. So that would be you are the F1 offspring of your parents. F2 is the second generation. So the offspring of the F1 generation. And then hybrids are the offspring of parents with different traits. you will need to remember these terms for a vocab test. So Mendel, what he liked to do was he crossbred true breeding. Remember, true breeding means one parent produces the offspring. So he crossbred true breeding tall plants with true breeding dwarf plants. So he crossbred these, and he noticed that tall and dwarf so a tall plant bred with a dwarf plant produced all tall offspring. That was kind of odd. So F1 generation, the first group of offspring from the original parents, were F1 hybrids, and they were all tall. Okay. All the offspring had the appearance of only one of the parents, which was the tall plant. The trait of the other parent seemed to disappear. So at the beginning, he thought that this dwarf trait was just lost. These plants somehow got rid of this trait. They kept the tall trait. So that was what he thought in the beginning. So he came to two conclusions here. He said that biological inheritance is determined by factors that are passed from one generation to the next, which is fairly obvious. And today we know these factors to be called genes. We know what genes are. We talked about those earlier. Each of the traits that Mendel observed, this was very, very, very important. It's controlled by one gene. It's hard to study traits that are controlled by multiple genes at different locations on the chromosome. But these two genes occurred in contrasting forms. For instance, one gene for tall, one gene for dwarf. So let's look at an example. The gene for height of pea plants occurs in tall and in dwarf form. So like I just told you, tall and dwarf, same gene but different form of that gene. And this different form of the gene is what we call alleles. There's that word again. That's going to be coming up quite often. So Mendel realized that some alleles were dominant. So if we look at this picture, it's pretty interesting. Seed shape. We breed a round seed plant with a wrinkled seed plant. We get a round seed. So if we look at this data, we can say that round is dominant. Here, yellow is dominant. Gray is dominant. Smooth seeds are dominant. Green pod shape or color is dominant. Axial flower position is dominant. And tall plant height is dominant. And all I did was look at the two breeds. So yellow mixed with a green plant, and we get a yellow offspring in the first generation. So that tells you that the yellow plant is dominant, or the yellow color is dominant. Okay, so this is the principle of dominance and recessiveness. And I want you to write this. It just tells us that some alleles are dominant, some is recessive. The dominant allele can cover up or mask a recessive allele. That is why when Mendel bred the tall plant with the dwarf plant, we got a tall plant because the tall plant was dominant and it dwarfed or masked, covered up the dwarf allele. So go ahead and write that for me. And I'll do the same. All right, so let's look at these definitions real quick. These will be on your vocab test. Dominant allele, if it is present in the offspring, the dominant trait will show up. So if there is one allele, remember each offspring has two alleles for each trait, but if there is one dominant allele, then that allele will show up. So go ahead and write that. And the recessive allele will show up in the offspring only if there are two recessive alleles and no dominant alleles. And we're going to talk about what this is called, but if you have two of the same allele, that's called homozygous. So you either have two dominant alleles, homozygous dominant, two recessive alleles, homozygous recessive. If you have two different alleles, so you have a dominant and a recessive allele, that's called heterozygous. Those are going to be two vocabulary words. So Mendel had another question. Had the dwarf trait, so had that trait that the dwarf plant had, the recessive allele, did it disappear? Or was it still present in the F1 offspring, just covered up? So what he allowed to happen, we're on page 4 now, is he allowed the hybrid, so the F1 generation, the hybrid tall plant, so the offspring that we got, from the first generation to self-pollinate. So we allowed this to pollinate itself. So we had two hybrid tall alleles. So we had an F1 tall plant mixed with an F1 tall plant, so first generation tall plant. Remember, it's hybrid, so this is coming from the tall mixed with the dwarf plant. And what he found was very, 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 very interesting. So we mixed, here's the first generation. Remember, don't get confused. Let's look at this picture. First generation. Let me see if I can draw on this real quick. I don't know that I can. But anyways, here's the tall plant, short plant. They mix. Remember, we got two tall plants in the F1 generation, if you look here. What happened was he let these self-pollinate, and he ended up with something miraculous. He ended up with 3 tall plants and 1 short plant. Very, very interesting. So go ahead and write this. He found that 3/4 were tall and 1/4 was dwarf. So 1 out of the 4 was a dwarf plant. Very, very interesting. So number 1 here, it's written for you. You found that 3/4 of the offspring were tall and 1/4 was dwarf. So that told him something. Evidently, the first generation tall plant that came from this P generation had to be carrying the dwarf trait. But in the tall plants, had to be hidden, because if it wasn't hidden, it would be short, or it'd be a mix. So the dwarf trait had been passed down to the offspring, and it reappeared in the F2 generation, so the second generation. Second generation offspring. Technically, this is generation 1, 2, and 3. We call them parent generation, first generation offspring, second generation offspring. So, question. Why did the recessive allele seem to disappear in the F1 generation and reappear in the F2 generation? We're on C. So Mendel realized that organisms have 2 alleles for every trait. And these 2 alleles are inherited, 1 from each parent. And if the offspring receives a dominant allele from one parent, the dominant trait will appear. The recessive traits only show up in the offspring if the offspring receives recessive alleles from each parent. So let's look at this real quick. Let's see if I can minimize this for a second. Let me see if I can find something to draw on here. So we'll just draw on, let me see here. It's been a while. All right, so we'll just write right here. So recessive, 2 R's, dominant, dominant, recessive, homozygous. If we have big R, little r, then the dominant trait will be there. If we have little r, so we're 2 recessive alleles, then we will see the recessive trait. So what happened here was we had this in the F2 generation. I know this is not perfect. We had big R, whoops, I messed up. Let me get my eraser. So we had big R, little r, big R, little r, R, R. So that was going to be the dominant allele, whatever, or the dominant trait, whatever that is. Big R, little r, still dominant. There's one dominant allele. Big R, little r, still the dominant allele. And then in this one generation, we have 2 recessive alleles. So if we look back at our paper here, let me figure out how to do this here. There we go. So if we look back here, this plant had 2 recessive alleles. All right, let's move on here. So this is number 2, C2. If the parent has 2 alleles for a trait, how does the parent pass only 1 allele to offspring? We know this. We talked about this. Okay? So this lies in type of cell division known as meiosis, which is the formation of gametes. Gametes, we know these sex cells, such as egg and sperm. Remember, meiosis results in, we go from 1 cell with 46 chromosomes to 4 cells of 23 chromosomes. So we split those in half. So we only get 1 allele each. Okay? So here's an example. So let's look here. So we're going to use this example with the capital T representing a dominant allele, and the lowercase t representing a recessive allele. All right, so during meiosis, we have big T, little t, replication, so we have 1 of each, or 2 of each, I'm sorry, so we replicate that. In meiosis, we have to replicate that. In interphase, we replicate the genetic material. Then after meiosis 1, we have 2 cells that are still 46 chromosomes apiece. In this case, we're just looking at 1 set of alleles here. And then second, meiosis 2 results in 4 cells with half the genetic material of the parent. Okay? 4 cells, half the genetic material of the parent. Okay? So during meiosis, the replicated DNA is replicated. That's during interphase. And we end up with 4 gametes. And this is how each parent passes only 1, notice 1 allele each to the offspring. Pretty neat stuff. All right, principle of segregation. That is what we just talked about. Okay, so this principle, discovered by Mendel again, says that every individual carries 2 alleles for the same trait. And these 2 alleles separate or segregate during the formation of sperm cells or sperm and egg cells. Again, meiosis, essentially. Okay? So now we're going to talk about homozygous, heterozygous. So don't get confused. Segregation is just a separation of genetic material. This is segregation, okay? Now, when they recombine, obviously they combine with another. If this is sperm, it would recombine with the egg, and then we would come back and have something that looks like this, okay? So homozygous, heterozygous, I already told you this, but I'll tell you again. So each offspring receives an allele from each parent. So each offspring, or let's take you for example, has 2 alleles for every trait. You got one of those from each of your parents, okay? Just like you get half your chromosome, you get 1 chromosome on, so you have 23 pairs of chromosomes. Each out of each pair, so say we take the first pair, you get half of that from your mother, half of that from your father, okay? So 1 chromosome each from your mother and father. Combination of alleles received by the offspring can be either homozygous or heterozygous. This is a very easy definition, guys. Homo meaning same, hetero meaning different. So, so 2 alleles are the same. Big T, little t, little t. Or heterozygous, meaning we have 1 dominant, 1 recessive allele. Very, very easy. Go ahead and write this down. I expect everyone to get that correct on their vocab test. All right, we're getting close to the end, guys. We're going to work through these Punnett square problems here in a minute, and we're going to finish on page 8 with moving on to dihybrid crosses and things, okay? So genotypes and phenotypes, I've told you this a lot, but I want you to write this down, okay? So genotype is the genetic makeup of the organism. Again, if you need to write this or pause this video, be my guest to do so. Genotype, again, the genetic makeup of the organism. Phenotype, think physical characteristics of an organism. Genotype makes way for your phenotype. Phenotype is what the organism looks like. Genotype is what causes that, okay? So, for example, let's look at Mendel's pea plants here. Specifically the tall allele, again, which was dominant over the dwarf allele. So here's your genotype, which are the alleles. Remember, alleles are found on the genes which code for specific traits. Again, we're talking about height here. Big T, so this is, I hope you know, this is homozygous dominant. Heterozygous, you don't say dominant or recessive, just heterozygous, meaning 1 of each. And then homozygous recessive, okay? And then, so this is the genotype. And then the trait, what we see with our eyes, is the phenotype. So big T would be tall. Big T, little t, heterozygous, is still tall because we have 1 dominant allele. And the only one that would be dwarf is homozygous recessive. All right. Now, the fun stuff. Let's start talking about probability. Please write this definition down. It will be in your or on your vocab quiz. But probability is the likelihood. We're specifically talking about genetics here. The likelihood that a particular event will occur. Okay, how do we know that? Well, we know that by looking at a Punnett square. Again, you're probably going to have to pause this video. I'm moving a little bit quicker now because some of this stuff is fairly easy. Just some definitions. And these Punnett squares are really easy as well. All right, so I want to put this up here real quick, and I want to go over this, okay? So we're going to look at a Punnett square, which is a diagram that shows allele combinations and what could result from a cross between 2 different parents. We always place the alleles of 1 parent on top and the alleles of the 2nd parent on the bottom. I'm sorry, on the side, left side that is. And then we mix those, okay? So we'll bring these 2 together, there and there. We put them here. Here goes here. Here goes here. There, there, then there. Okay, it's pretty simple. I think you'll understand it. I'm actually going to do this with you. Let me make this a little larger, and I will draw on this for a second. So we go big T, big T. Big T, little t. Again, I'm bringing this here, this down, this goes down, this goes down, this goes across, this goes across. Just like that. So we have homozygous dominant, heterozygous, homozygous dominant. All of those are either homozygous dominant or heterozygous, still with one dominant trait. Therefore, they are all tall. And then actually, I apologize. This is also heterozygous with one dominant trait, so therefore it is also tall. All right, moving on. Fairly easy. Please pause this video and do this by yourself. So pause this video and do this by yourself. I'm going to do it with you. So we have the genotype. I want you to pause it and do it, and then you can come back and get the answer, okay? So I want you to try these by yourself. So first parent, let me write on it real quick, is big T, that's here. Second parent is small t, small t. We bring these across. Always put the recessive trait on the right-hand side. Bring this down. So we have 4 heterozygous plants. All will be tall, right? So let's look and see if that is right. All right. All of these will be tall because we have one dominant allele in each. So I want you to use this table to give the probabilities of the genotypes. So again, this is pretty easy. We had 4 heterozygous genotypes, big T, little t, and 4 tall plants. All right. Let's look at the F1 generation, self-pollinating. So remember, self-pollinating, he let those heterozygous plants self-pollinate. So we would have big T, little t mixed with big T, little t. That's from the last page. He let them self-pollinate. So one parent goes here, big T, little t. The other parent goes here, big T, little t. Bring them down, big T, big T, big T, little t, big T, little t. Remember, big T, little t. Little t, little t. So let's look at our genotypes here. Homozygous. It's going to be tall. 2 heterozygous, which will be tall because we have one dominant trait. And then we have a recessive trait. First one, and this would be dwarf. All right, hopefully that's what you got. If you have any questions, please let me know. Good. All right. Couple more, 3 more. I want you to do these by yourself. And I want you to, so it's asking you to show all your work, Punnett Square, summarize your findings in the table. So pause this video now. Go ahead and do that. And I will show you how I would do it. So we have a dominant, dimples is dominant over the absence of dimples, cross a heterozygous dimpled man. So we'll do big D, little d. Remember, heterozygous. With a woman who does not have dimples. If she does not have dimples, remember, she's the absent of dimples is recessive, so she must be little d, little d. So we have big D, little d, little d, little d, big D, little d, little d, little d. So our chart, genotype, phenotype. We have, let's do this. 1/2 big D, little d, and 1/2 little d, little d. And this goes to tall, half, and oops. And we're not talking about tall anymore, are we? So dimples, sorry, this is a mess. I'm trying to write this with my mouse. And 1/2 no dimples. You get the picture. So again, just what I did. You can pause this, look at it. 2/4 is 1/2, so don't get confused. So 1/2 dimples, 1/2 no dimples. Okay, white squirrel albinism. So pause this and go. Do it yourself. Pause it. Pause it. Stop. Don't keep watching. I'm going to solve it now. Heterozygous, heterozygous. Homozygous dominant. 1 heterozygous or 2 heterozygous traits or alleles. And 1 homozygous recessive. So we should have 3/4 should be, let's see, normal skin's dominant, so it should be normal skin. Again, 3/4 are all of these. So 3/4 normal, 1/4 albino. If you have any questions, let me know, please. There you go. Last one. Pause. Do this one 100% on your own. Go. All right, so it tells us the allele for short hair is dominant. The allele for long hair is recessive. 2 short-haired dogs have a litter of puppies. Some of the puppies have short hair. Some of the puppies have long hair. So that tells us there has to be a recessive trait in here. But both dogs are short-haired, so they are dominant. So both dogs have to have the dominant trait. But the only way, if they have one dominant trait and they're short-haired, the 2 short-haired dogs, the only way they can have short-haired puppies is if they are heterozygous. Because, oops, I don't know why I wrote R. We're going to change this to B. If one of these was homozygous dominant, then all of these are going to have a dominant allele. But here we have B, 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 little b, little b. Genotypes. 1/4 B, B. 2/4 heterozygous. 1/4 homozygous recessive. So these two will give us 3/4 short hair. This one will be the only one that gives us 1/4 long hair. There's your phenotypes. So let's close this out and look at this one more time. All right, so here's a good question. If the litter of puppies contained 12 pups, how many would you expect to have long hair? Well, here we have 4, and out of the 4, 3 of them have short hair, 1 has long hair. So we have 4 here. So how many times would we have to do this to get to 12? 4 times 3 is 12. So take 3 of these. So 3 times 3 is 9 twelfths. And then 9 twelfths, 3 times 1 is 3 twelfths. So we would expect 3 puppies to have long hair and 9 puppies to have short hair. So there you go. And 3 pups with long hair. There we go. All right, so we're going to stop there. And your assignment right now is monohybrid mice. I want you to do this assignment. Okay, work this assignment. Now, you will not have the answer key on there, but I want you to work this assignment by yourself. And then once you get done, you have one other thing to do, and that is the monohybrid worksheet. Looks difficult, but it's not very hard. Okay, so do both of those. When you get done, you can move on to the second section, starting on page 8, independent assortment, and we're going to go. We're going to look at dihybrid crosses, and we will stop there on page 11. All right. I hope you enjoy this new way of learning. I know this was a fairly lengthy PowerPoint. We're going on 52 minutes now, but you can put this on YouTube in half speed or double speed or what you want to get through it a little quicker. And this is going to be some self-paced learning, so those of you that like to go ahead and do things quicker, you go ahead, move as fast as you want to. I'll give you the vocab test and the unit test as soon as you get done with everything, okay? So there's monohybrid. We're going to go to dihybrid next. So if you have any questions, let me know.